3, 2, 1.
The biochemical test. First, sterilize the inoculating needle. Pass the agar plate onto the flame to avoid cross-contamination. Then choose and take a single colony. For the TSI, stab and then streak. For the LIA, stab and then streak. And for the citrate, streak only. Lastly, for the SIM, use stab only method in upright position. AST or the antibiotic susceptibility testing. Prior the procedure, always begin with labeling the tubes and the Mueller-Hinton agar plate. Sterilize the inoculating needle till red hot. Using a sterile dropper, transfer 3 ml of NSS into the tube. Then, select a pure culture from the TSI. Aseptically emulsify a colony in the sterile saline solution. Mix it thoroughly to ensure that no solid material from the colony is visible in the saline solution. Repeat until the turbidity of the saline solution visually match that of the standard turbidity or the McFarland. Take a sterile swab and dip it into the broth culture of the organism. Gently squeeze the swab against the inside of the tube in order to remove excess fluid in the swab. The swab is streaked in 3 directions over the surface of the Mueller-Hinton agar to obtain uniform growth. A final sweep is made around the rim of the agar. Allow the plate to dry for 5 to 10 minutes. Using the sterile forceps, place paper discs impregnated with a fixed concentration of an antibiotic on the surface agar plates at equal distance. plate will be incubated for 24 hours at 37 degrees Celsius.
So, Vitek machine is a fully automated machine, no? It can identify bacteria and yeast. Nasa specific test tube. Pwede siya plastic, pwede po siya kanang polystyrene na kamot sa itsura. Pero siya lang yung gamit sa lang. Okay? Tuloy po siya natin. Okay? So, suspension, 3 ml dapat. So, since natin kanti guys, so 3 ml, mapunta sa 3 ml. Sapa lang, then dispense. I-close siya para di mapunta. And then guys, as you can see, naman siya ang Vitek cards. So, ang Vitek cards, it consists of 2. For identification and ESP. So, kani siya, Pokemon cards. Oh, kani. Tama ni. ESP. So, kani siya for antibiotic. Wala ka nag-meller into agar ani guys. Pero kani lang sa card. Then, kani siya, identification for gram negative. So, kani siya guys. Kani siya for identification. It consists of 64 wells. 64 sa mark. Actually, nagkakit kayo biochem test, coagulase, catalase, etc. and so on. So, ato ang sa lab is 5 lang ginagamit for gram negative and sa gram fast, catalase, coagulase lang. So that's why it can differentiate or speciate bacteria na grabe ka rare na bacteria. Kaya niya mo identify. Guys, kung siya tinatawag na transfer gene. Bawal na siya i-touch. Okay? Bawal siya makuntang. Okay? So, if wala ka mo nag-start, ano lang sa? Ano ba? To prevent what? From touching or accidentally touch ang transfer gene. Kasi mag-ibigay po yan. Okay? Then, pakitaan mo sa ESP. Siyang ESP. Wala siya color. Siyempre, kaya antibiotic lang may sila. Okay? Then, as you can see, no, nasa mga, what? Barcoded din siya. So, since barcoded siya, dapat ang ang laboratory is naka-LIS. Blue, ang transfer tube for identification and gray for ESP. Then, wala yung mga color, di ba? Antibiotic naman. Wala. Then, hindi na siya gram net, gram fast. For example, makon kita makuha. Okay? So, kung makon kita makuha, since makon kita makuha, gram negative na, what? Vitek card atong gamiton. Since gram negative na Vitek card atong gamiton, so kani atong gamiton for transfer sa suspension diri sa AST. Ingay mo siya. Sa identification, magbuhat ka og suspension. Pag makabuhat ka og suspension, dili na ka magbuhat og suspension diri. So, for example, 3 ml ni siya. Let's say 3 ml. Dili na ka magbuhat og suspension. Atong buhaton, all you have to do is to transfer. Kani ko ang ani type it transfer nimo ni suspension diri then i-dispense. Bota suspension kasi pagbuhat ato same lang gyapon sa unsa. Katong dati, katong same lang gyapon sa buhat ko. Suspension sa meron to agar. Sir Mark, magamit ang backfire dili na. Ato gamiton is kani. Tingnan na sa inyo. Then this is called as density check, no? Gina-check niya ang optical density sa suspension. So, in short, gina-check niya kung sa ka-turbid ang suspension. So, kuha tanong. Wait lang, tanong na po. See? Touch lang. 1, 2, 3. Then, i-mix natin siya. Before i-mix, syempre. So, since wala tayong vortex, kamot lang sa, no? Sa kung naman ko tayong vortex, nauna na siya. Din ang vortex na mag-mix para homogeneous siya. Nagulat ang ganda. Sige guys, natuwa sa siya. Siyempre, kung may mga markings, abis sa computer, kani, one siya, ganun, passenger DDT. So, usap pa ko on sa markings ko nga. Then, para ma-zero siya, tingnan lang. Okay? Sino is on? Zero. Kani zero para ma-zero siya, okay? Wala siya pair. Isa niya pair, yes. Then, siyempre, yung sabi glass plastic. Glass, dapat drag glass, nakakaro sa glass. Para na siya yan, no? Ituyok siya siya 360 degree. Okay, amot ganun to siya. Amot to siya. No? Then, pindutan niya siya. Then naman yung maragam. Click. 0.40. Dili, di ba? Ganun dili ba?
0.5 to 0.63.
Yes, tama. 0.5 to 0.63. Tapos, then, then, wala siya there. 0.47. Doon, kapit na to. So, asa yung hindi ka pa nakakuli kasi. So, asa yung mag ay. Grab name, di ba? Yes. Because, blue. So, ako ay. Oops, wala yung bobuski. So, diretso lang siya. Kailangan may mix na mark, no? May mix yan. Machine na mag-mix yun. Okay na? Prepared na. So, pwede na ito sa machine. Wait lang ha. Maluwas na ito yung frame. Susunod dito. So, uy, bones. Oh, 3.9 million.
Sir, 19 minutes na yung mga answer. Oh, screen time. Hawa sa mudra. Ako ang video nakasanalay.
So, yan guys. Video na siya. Teaser lang. Teaser lang ako sa video. So, before pa na siya isulog guys, dapat i-transfer na siya yung ganyan transfer queue. Okay? Ganyan transfer queue. O, diba? Oops. Okay. Okay, ano na. Then, pagsulod, ano naman yung arrow? Anong arrow?
Oh, na arrow.
Check. Then, close. Then, saan ba niya siya?
Isunod ka ng cellphone para ma-video niyo.
Dara, start fill. Okay? Complete naman guys. So, kani? So, i-transfer. Complete din yung karami.
22 minutes na.
Then, yan eh. So, as you can see, isubsub na ganun.
Uy!
Tapos, ano ba? Color change. Then,
Sir, di na maklaro.
then, balik na ito diri ha.
AFB staining. First and foremost, label the glass slides. Write down the yearly serial number or order number of sputum specimen on an end of the slide. Ensure complete confidentiality of test. Pass the glass slides into the flames to avoid the cross-contaminants. Using a sterile swab, fish out a loopful of yellowish particles of sputum. Spread one loopful of the sputum evenly on a clean labeled glass slide, approximately 2 by 3 cm in size. 2 by 3 cm size slide approximately has 300 OIO fields. Repeated coil type of distribution is used to evenly distribute the bacilli, if present. Then, allow the smear to dry completely at a room temperature for 15 minutes and fix it by passing through the flame 2 to 3 times about 2 to 3 seconds each. Place the slide in the staining bridge. Using the Ziehl-Neelsen method, pour carbol-fuchsin stain to cover the whole surface of the slide. Then, heat the slide until steam comes off the stain. Do not boil and do not allow the slide to dry. Leave it for 10 minutes. Tilt the slide to drain off excess stain. Wash the staining solution off with a gentle stream of running water. The next step is decolorization. Pour the whole slide with acid alcohol or ethanol and leave it until no more color drains from the smear. The exact time of application, just make sure that no carbol-fuchsin remains. After the application, wash the slide once again. Counter staining. Pour methylene blue stain to cover the whole surface of the slide and leave for 15 to 30 seconds. Pour off methylene blue stain. Wash the slide with a gentle stream of running water. Drying the stained slide. Tilt and place the slide on the slide rack to air dry. AFB reading. For AFB reporting, grading is 0 when no AFB seen in 300 visual fields, plus N if 1 to 9 AFB in 100 visual fields. Example, plus 1 or plus 2 depending on how many AFB counted. 1 plus if 10 to 99 AFB in 100 visual fields. 2 plus if 1 to 10 AFB per OIF in at least 50 visual fields. 3 plus if more than 10 per OIF AFB in at least 20 visual fields.